Hello students. Okay, I hope all of you are doing fine. All right. So today we'll study about the cell cycle and division. So cell cycle and cell division is a very important topic. Okay, and it is the topic which forms the basics of molecular basis of inheritance, which we study later in our biology. And this, while it is very important, it's also interesting as well. Okay, so I want you all to pay attention to this class, and let's maintain this class active and fun so that each and every topic we understand it carefully and significantly. All right, okay. So we'll begin the class. So cell cycle and cell division, how do we define it? So what is cell cycle and what is cell division? Why does it has a significance? Okay. On a daily basis, whether it is an animal cell or a plant cell, okay, it keeps dividing. Okay. There are particular kinds of cells that keep dividing in plants. Meristematic cells are there. There are meristematic cells that do keep dividing. Okay, and in animals like us, in human beings, there are epithelial cells. Okay, that do keep dividing on a regular basis. Okay, so while it is important for growth, it is also important for repair. So what are the two important parts of cell cycle that we need to remember? Growth growth and repair. Understood? So growth and repair are one of the most important parts of the cell cycle. Okay, so that's how we have to remember this. Okay, so now while we have discussed now, let's begin with the understanding the depth of the cell cycle. So cell cycle has all these phases, okay, in which the G1, S and G2 are together known as interphase. So we can divide cell cycle into two parts. What are those two parts? Okay, what are those two parts of cell cycle? First, we can divide it into first two parts is, okay, that is interphase, and that is another one is M phase. So M phase is the actual dividing phase, which is also known as mitosis. Okay, and interphase is the phase in which the cell takes time to grow. Okay, and make up all the components that are required for the cell division that takes place in the M phase. Okay, so interphase is further divided into three phases. What are those three phases? They are G1, S, and G2 phases. See here, as soon as this all, why do we call it a cycle? What is the difference between a pathway and a cycle? In a pathway, there is a first product and the last product, and they don't continue as a cycle. But in a in a cycle, if we see, unlike a pathway, okay, they keep interchanging. See here, after the M phase, G1 starts, then S phase comes, then G2, then again M. And this keeps on going unless there is an exception of G0, which I'll explain later. Okay, so in this gap 1, which is known as gap 1, okay, or G1, okay, in this phase, the cell takes time or a gap, and after that there is no DNA replication in G1 phase, and later in S phase there is DNA replication, which is a very important part of interphase. DNA replication. So in what happens in DNA replication, the amount of DNA gets doubled because see, in M phase the one cell divides into two cells, right? One cell divides into two cells. So when one cell is dividing into two cells, it has to equally distributed DNA content to each of these daughter cells. So when it has to do that, it has to have the double the content of the DNA so that it can distribute it to the daughter cells. So if you have studied your NCRT, it has clearly given if you have a 2C content of DNA, before the S phase. After S phase, you'll have, after S phase, how much you'll have? 4C content of DNA. Alright. So that's how you actually denote the S phase and denote the DNA content. But the chromosome number, does it change or it doesn't change? It does not change. The chromosome content would not change. The chromosome content will be the same. Okay. So if we have 2n number of chromosomes, so after S phase it will have same 2n number of chromosomes and there will be no changes there regarding the chromosome content. Okay. Now if we take the G2 phase, what happens in G2 phase? If we take the G2 phase, what happens in G2 phase? In G2 phase, you will have protein formation. This protein formation, why is it necessary? Protein formation is necessary for all the components that are actually forming during the cell division, like microtubules, spindle fibers, and everything. Added to this, in animal cells, there is a special thing that also gets done during the S phase. What is that special thing that gets done during the S phase? During the S phase, there is another thing that happens in animal cells. In animal cells, the centriole also divides or duplicates for each pole. Two poles are there. So one centriole will be there and it will duplicate for each of this pole so that the spindle fibers can align to it and bring the chromosomes to the each pole in this cell division. Okay, we'll understand the significance of centriole further. So one last thing is the G naught. What is the G naught phase? This is known as the quiescent cell stage, in which after the G1 phase, the cell enters into G naught and it doesn't undergo any cell division further. It doesn't undergo the interphase further and it doesn't enter the M phase as well. So the reason for behind this is the cell is not asked to do so. So nothing is happening, or any of this is not happening without any command from the cell. So the cell right now doesn't want to divide. It does not have to divide or it doesn't want to divide, and until and unless it is asked to do so, it won't. So in this phase, it will stay like the same and it won't divide further. This is known as the G naught or the quiescent cell stage. Alright, okay, so we'll enter into the next part of the lecture, that is M phase, which is the one most significant part of the cell cycle and cell division, right? Okay. There are some of the points which we have to remember with respect to the cell division. And what are those points which we need to remember? Those points are: number one point is every 24-hour cell division takes place once in 24 hours. Okay, this refers in organisms like in yeast, and every 90 minutes it takes place. Okay, so that's one difference. This is the regular time which takes place in, like, in us human beings. Okay, then number two, okay, 95% of the cell cycle is taken by interphase only. Only 5% is taken by M phase. So this has to be clear for all of you. Okay. All right. So now this is enough. We'll move on to the M phase now. So in M phase, okay, there are they can we see M phase is the phase in which the cell is dividing. Okay, and we can't draw always the clear cut lines, but due to so that we can understand each of the event which is happening. Okay, so we conveniently divide them into prophase, metaphase, anaphase, and telophase. Prophase, see, prophase, metaphase, anaphase, and telophase. All right. Okay. Now we'll understand each of these phases. So what happens in prophase? As we already studied, okay, that there is a DNA replication that takes place in S phase. So it happens in the form of a thread like this. This is a thread, chromatin thread. Now this chromatin thread is completely entangled with each other. Okay, it's not properly aligned with each other. It is completely entangled. And now it is important that this chromatin fibers are condensed to form chromosomes. So it is very important that these chromatin material condenses and it forms chromosomes. Okay. So these chromosomes, number one, what happens in prophase? Chromatin condensation takes place. Chromatin condensation to form chromosomes. Okay. This is the first thing that takes place. Number two, okay, now this has happened. Another important thing that takes place is formation of the initiation of the mitotic spindle. Now, mitotic spindle is very much important. The spindle fibers, the microtubules, all of these proteinaceous elements which were formed in the G2 phase as we remember. Okay, so initiation of spindle or mitotic spindle. Okay, so mitotic spindle. Yeah, this is another important event that takes place in prophase. And the last is, which is also very important. What is it? Okay, the cytoplasmic components such as Golgi complex, okay, endoplasmic reticulum, mitochondria, and also the nucleolus inside the nucleus. Okay, all of this disappear so that enough space is given for the cell to divide. Okay, so that all of these organelles are disappeared in this phase. Okay, so what are the main events that takes place in prophase? Chromatin condensation takes place to form the chromosomes. Okay, initiation of the mitotic spindle takes place, and number three, the Golgi apparatus, endoplasmic reticulum, mitochondria, and nucleolus, they all disappear so that enough space is given for cell division. Okay, and also the nuclear membrane which is surrounding the nucleus starts disappearing slowly, and this actually takes place finally in metaphase. See here, see each of these diagrams signifies properly. See, the chromatin thread in the early prophase is converted into chromosomes after condensation. Okay, now after condensation, see the nuclear membrane is disappearing here. You can see here the nuclear membrane is disappearing, okay, slowly dissolving, and it has finally completely dissolved in the metaphase. So metaphase begins with dissolution of nuclear membrane. Okay, and the chromosomes take proper shape, and after this shape is taken as well, they form sister chromatids are formed actually. Sister chromatids. What are sister chromatids? They are part of a same chromosome. Two parts of a same chromosome. See here. We can see here. If we see like this, see. This is the chromosome. So this is one chromatid and this is another chromatid. Okay. These both are known as sister chromatids. This is the centromere. Okay. All right. So these are known as sister chromatids. Okay. So sister chromatids form. Okay. And number three, the spindle fibers attach themselves to centromere. They are coming from the centriole here. This is the centriole, and they are attaching themselves to the centromeres. See here, these are the centromeres. These are the centrioles at two poles, and these are the centromeres of each chromosome. Okay, so they attach themselves to the centromere, and last and the final one, which is most important one, is formation of metaphase plate. Okay, so that is what takes place in metaphase. Dissolution of nuclear membrane, sister chromatids form, spindle fibers attach themselves to the centromeres and formation of metaphase plate takes place, in which the chromosomes will align in the center of the cell. Okay, they properly align in the center of the cell. Now this is where actually the chromosomes start separating. Each of the chromosomes will start separating with each set of sister chromatid going to the opposite poles. Okay, in anaphase. So anaphase is marked with, okay, separation of chromosomes, separation of sister chromatids, we can say, right? Let's say separation of sister chromatids. Separation of sister chromatids. Okay, then the centromeres are splitting. So centromeres split. Okay, an interesting part here. See, the centromeres are being carried away by the spindle fibers, and they are facing the poles, and these arms of each of these chromosomes are trailing behind them. Okay, so this is the middle of the cell, and from this middle of the cell they have started going towards the poles already. Okay, they were along the centromere plate here. If you can see here, they were along the centromere plate here, and after the centromere plate, okay, metaphasic plate, they have now started traveling to the poles due to the spindle fibers. The spindle fibers are pulling them to the poles. Okay, so the centromere split. Okay, and this is the event that takes place finally in anaphase. So finally in telophase, what happens in telophase? Okay, so anaphase is a very small event, right? So in telophase, these chromosomes have already traveled to their poles, and they dissolve, and they lose their discrete structures. So chromosomes lose their discrete structure. Okay, chromosomes, they lose their discrete structure. You won't see a proper structure here. Number one. Okay, number two, the nuclear membrane starts appearing again. Appears again. Okay, see, this is the nuclear membrane around it. Okay, then number three. Okay, all those organelles which we talked about, which they disappear during the prophase, they will appear again. Those are Golgi, okay, ER, mitochondria, and last one is nucleolus. These are some of the organelles which we actually significantly we should remember. Okay, now the telophase is done. The cells have to actually form. We need to see two different cells finally, and till now what has happened, only the nucleus has separated, which we will say it as karyokinesis. Karyokinesis. What is karyokinesis? Karyo means nucleus and kinesis means movement, and this means that the nucleus has separated. Nucleus separation or division. Division of nucleus. Okay, very important. This term is very important. You have to remember the meaning of it. Now, once this has done, we come to the last part. What is the last part of this? Is the cytokinesis. As mentioned, karyokinesis is the separation of nucleus. Cytokinesis is the separation of cytoplasm. Cytoplasm separation. All right, so now the cytoplasm gets separated. Okay, and there are two ways it can happen according to each cell. How do we say this? See, look here. In animals, okay, we'll go a little bit up. Yes, okay. See, this is the way how it takes place in animals. That a furrow takes place in this is what takes place in animals. In animals, it is initiated by formation of a furrow. in the end, formation of furrow at the end of the cell, and this travels all the way back to the center, all the way to center. This furrow all the way, it will travel here like this in this direction, okay, and it will finally divide the cell into two parts, okay, and it and divides cell in two parts, okay. Now this is what happens in animals. There is another way that takes place in plants. Now plants have a cell wall and this can't take place. So in plants what happens is, okay, the furrow will begin from here, okay, and it will travel the opposite side towards the outside, and then the plants will separate, and then the plant cell will separate and it will form two daughter cells in the end because they have a cell wall. So in plants it begins furrow at the end, right? So the furrow is in from begins from the center. Furrow begins at center, travels all the way to the end, and then divides the cell into two parts. Two cells. This is also into two cells, not two parts. Two cells, okay. All right, so this is finally cytokinesis, okay. Now we have to finally summarize the mitosis. So how do we summarize mitosis? How do we summarize the cell cycle and cell division which we studied till now? Okay, the cell cycle and cell division is an important part and an integral part of a cell. Okay, in plant cells, mainly meristematic cells are the examples, and in animal cells, epithelial cells are the examples which I have given. Okay, they're important for growth and repair. Okay, and it has two phases: interphase and M phase. Okay, the interphase is again divided into G1, S and G2, in which S phase is very important because it has DNA replication taking place in that. And the M phase is being divided into four phases, that is prophase, metaphase, anaphase, and telophase. Prophase, condensation of chromosomes and disappearance of organelles takes place along with formation of mitotic spindle. Okay. Then in metaphase, the nuclear membrane will disappear, the sister chromatids will appear, the spindle fibers will attach themselves to the centromeres, and the metaphase plate will form on which the chromosomes will align properly, okay, in a straight line. Okay, and then in the anaphase, it's a very small phase in which the separation of the sister chromatids and the centrome takes place, and these centromeres split, and these chromosomes travel to opposite poles with the help of spindle fibers. Okay, last in telophase, okay, these chromosomes lose their discrete structures on the shape. Okay, and the nuclear membrane reappears again, the Golgi complex, okay, endoplasmic reticulum, mitochondria, and nucleolus, they all reappear again. Okay, and finally these all phases which we talked till now will come under karyokinesis, that is separation of the nuclear part of the cell. Okay, now cytokinesis has to take place, that is the separation of cytoplasm has to take place so that the cell is completely divided, and that is what is known as cytokinesis, which we discussed, which is different in each animal and plant cell. In animal cell, okay, formation of furrow takes place in the end, travels to the center and divides it into two cells. In plants, because of the cell wall, the furrow starts in the middle and travels towards the end and divides the dividing cell into two cells. Okay, so mitosis is very important part of cell division, and I hope you all understood what I tried to convey to you, what I tried to teach you. Thank you for giving me this opportunity. Have a nice day.